some may not have the right chapter in your actual syllabus, but nonetheless, this is going to be our GMO reference lecture. So I'll put GMO ref down here. It's a really good one. So you can look in your book as well. This is a good place for it, but Hawaii's papaya industry seemed doomed in just a few decades ago, and so a deadly pathogen called the papaya ring spot virus had spread throughout the islands. It appeared to completely decimate the papaya plant population. This is going to tie in, I swear. So scientists from the University of Hawaii were able to rescue the industry by creating a new genetically engineered papaya ring spot virus resistant strain of papaya. So today the papaya industry is once again vibrant, and the vast majority of Hawaiians' papayas are actually genetically modified organisms. So there's a little tidbit for those of you guys who are doing papaya. And for those of you guys who aren't, this is kind of the purpose of our posters, is to be able to look at some of these facts from other people, and that there are purposes for our genetically modified organisms. And that's just one. So some critics have raised some safety concerns for the people who eat them and the environment, which is fine. You should definitely be researching that. Should we in fact be concerned about the safety of GMO crops? So the question continues to foster considerable debate and disagreement. And it's not wrong to have an opinion on these. I will say that. The opinion part is fine, but we want to just give the facts about our particular GMO. So we do have some GMOs in our diet, but DNA technologies affect our lives in many other ways. So there's some gene cloning, gene profile, or DNA profiling, some new technologies, and some other questions we can investigate. Unfortunately, we don't get to go into all the DNA technology stuff, which is fascinating. If you have any questions on that and how they do DNA profiling for like criminal justice, if that's something that you want to get into or look at forensic stuff, I'm happy to show you and point you to those references in the book because they are fascinating. But at least we'll kind of get to it. So if you don't know what papaya looks like, it looks like this. It's a fruit. And this is seeds in the middle. Okay, so we're going to maybe look at gene cloning and genetically modified organisms, though I will tell you that DNA profiling and genomics are a part of Chapter 12 as well, but we're only going to look at the first half. So gene cloning, the whole idea is that I want to give you a way to see this, because this is sort of the creation of section, is you may find that there's some wording in here that's similar. But as I said before, if you find a link that works better or that has the full description, but you're not really sure what it's saying, you're welcome to email it to me, and I'll put it into non-scientific, regular human terms with you. Okay. So biotechnology, it's the manipulation of organisms and their components to make useful products. So for thousands of years, humans have used microbes to make wine and cheese, to breed stock and dogs and other animals. This is actually what we'll get to with evolution at the start of next semester. This is the basis for that. But DNA technology is a set of modern laboratory techniques used to study and manipulate genetic material. The manipulate part just means to kind of manipulate actually means to change by hand, so it means that we're going to kind of alter it by hand. So genetic engineering, one of those terms we need to know, involves manipulating genes for practical purposes. So practical purposes just means that there is a reason for it. It's not just like, oh, we just started messing with genetics because we wanted to experiment. It means we actually had a reason for it. So there is a reason to do genetic engineering. We're not just making Frankenstein's monsters over here. We have a purpose for it. Not just like, we want to create new life and put organisms together. It's like, no, we're trying to solve a problem. It includes the glowfish. So if you have glowfish, that's one that's supposed to be involved in it. There's a whole environmental section on that it was created for environmental detection. So look into that one. If I can spell on the screen, there you go. So it had an environmental purpose. There is a practical purpose. Same with glow cats. If your group hasn't done glow cats, ask me about that because I'll show it to you. So gene cloning leads to the production of multiple identical copies of gene-carrying pieces of DNA in vitro, which means a test tube, to form a single DNA molecule. So you take one gene and then you just kind of repeat it over and over again to make a single DNA molecule. Okay. We also have recombinant DNA. It's formed by joining nucleotide sequences from two different sources and often different species. So this is kind of the crux of our GMOs, that typically they're going to involve different species. So one source contains the gene and the other to be cloned. Another source is a gene carrier. So it's going to carry around our gene and actually put it in there. That's what we call a vector. Plasmids are small circular pieces of DNA that replicate separately from a much larger bacterial chromosome, so they're used as vector. So if this is our bacteria, we've got this whole it's one big circular piece of DNA in the middle. Remember, they don't have a nucleus, but they also have these separate little pieces of DNA. Those are the plasmids, and those get used a lot to carry genes that we're interested in applying to different species, if that makes sense. So the following steps are used in cloning a gene, just so you can see that, is that we're going to isolate one of those plasmid DNAs. The DNA containing the gene of interest is isolated, so basically you just get those things alone. The plasmid DNA is treated, so that's our bacterial DNA, is treated with what's called a restriction enzyme. They're like little DNA scissors. I'm going to draw you little scissors. It looks like a bow. Is that better? It's a little better. Okay. Scratch the bow. Get the scissors. So it's like a little DNA pair of scissors. And it cuts in one piece, opening the circle. The DNA with the target gene is treated with the same enzyme, the same exact pair of scissors. Comes down here, does a little bit more cutting. And the plasmid and the target DNA are then mixed together and they associate with each other. Recombinant DNA molecules are produced when a larger enzyme, so the DNA ligase that we saw before, the little DNA, let me see if I can draw you a glue bottle. Not well. But anyways, it's a glue bottle, so it's our DNA ligase will join all those segments together because it's sort of like a little DNA glue. So it's a really simple kind of concept, right? Recombinant plasmid and the target gene are taken up by the bacterial cell, and then it actually reproduces because it undergoes that whole binary fission, mitosis type things, making identical cells. But the idea is that you take the thing that you're looking to put it in, you take it from the segment that you're looking for, and you use the same sort of pair of scissors to cut through each of them. Let's see if I can make better scissors. But you use the same set of scissors to cut each of them, and then you use a ligase and just glue all those pieces back together. That's more or less it. So here's our plasmid. We isolate it. Here's the particular sequence that we're interested in. We're going to cut it with that enzyme, or what's called a restriction enzyme. Then we put it together because we have these fun things called sticky ends that actually will complement each other, and put it there. And then you get it in there, a DNA ligase will come in and glue all the pieces together, and then you put it back in your DNA, or in your bacterium. The bacterium kind of goes through and makes a whole crap ton of copies. So then you've got a plasmid that has the gene that you're looking for, that you can then apply it to other kinds of things. Okay, so some of those things that you can apply it to is like a gene for pest resistance is inserted into plants. So that might be some of your pest-resistant situations. Maybe we have a gene that's inserted into a bacteria that can help us clean up some toxic waste, which, if I remember correctly, there's been a whole spillage of oil in an area. So this might be a thing that we need to look into again. All right. So some of these things, to go on a little bit further, those restriction enzymes also are kind of little DNA scissors. Just going to keep drawing you crappy scissors and deal with it. So they recognize a particular short DNA sequence called the restriction site. They'll cut both strands of the DNA at precise points in the sequence and yielding pieces called restriction fragments. So it's just like taking a strip of paper and it's like, I need you to cut it here and here and here. I need you to cut it into fourths. Well, then there's like a particular way that you measure that out and you do it. So restriction enzymes are very precise. That's the point. Very precise. Once the cut, we can paste them back together with our DNA ligase, our little glue. like a dirty glue bottle. So here's sort of what it might look like. Here's our restriction site. We'll apply what's our restriction enzyme. And notice it cuts it so we only have like half of it open. That's what we call a sticky end. So when we cut it in this way, it kind of cuts in a weird pattern, specifically so that way we can insert our gene of interest with some sticky ends as well. You put those together, they match up nicely because we've used the same sort of restriction enzyme scissors on them. And then we get our DNA ligase to come in and glue everything together. So more or less, it's fancy terms. Restriction enzymes and DNA ligase are really specifically for cutting and pasting. That's it. That's a super simplified version. So we get a little idea too of some other stuff, because it would be a good time to break if you needed to, but here's another definition. So we have a genomic library, which is a collection of all the cloned DNA fragments from a target genome. So that's kind of the genome's all the DNA that we would find in an organism. So genomic libraries can be constructed with different types of vectors. In a plasmid library, genomic DNA is carried by plasmid. In a bacteriophage, or that's a little is a little virus, in its library, the genomic DNA is incorporated into the bacteriophage DNA. Some other things is that we do have things called probes, which bind very selectively to cloned DNA. So probes can be DNA or RNA sequences that are complementary to a portion of a gene of interest. A probe can bind to that interest by base pairing, and probes are usually labeled with some radioactive stuff just so we can find it. That's all. Again, I'm giving you some background, so in case you saw any of those kinds of terminology in your creation of areas, you would sort of know where to go for it. So, well, let's reference. Here's a little bit more specifically to our genetically modified organisms. So recombinant cells in organisms constructed by DNA technologies are used to manufacture many useful products, chiefly proteins. Bacteria are often the best organism for manufacturing a protein because bacteria have those plasmids that we talked about, and we have some phages or some viruses that infect those bacteria available. We can grow them rapidly and they don't cost a whole lot. They can be engineered to produce large amounts of a particular protein, and they often secrete the proteins directly into their own growth medium, which is handy because they just kind of the bacterial equivalent of just poop out the target protein. It's like, well, that's handy. I will say, though, for bacteria, it's usually E. coli because they know the whole genome, like all of the DNA sequence in it. And even the non-virulent type of E. coli just smells terrible. Like, it's got a very distinct smell, and it's awful. You have no idea. All right, we can also sometimes use yeast cells as what our vector is, which is, you know, housing the DNA. So these are eukaryotes. They're super easy to grow. They've long been used to make bread and beer. So they can take foreign DNA and integrate it into their genomes. And they're often better than bacteria at synthesizing and secreting eukaryotic proteins. So sometimes we use the yeast cells because they're a fungi. And they'll work through those things. But it's not going to work all the time. So mammalian cells must be used to produce glycoproteins, proteins with a chain of sugars attached. They include human erythropoietin, which is EPO, which stimulates the production of red blood cells, factor VIII to treat hemophilia, tissue plasminogen activators used to treat heart attacks and strokes. So you can't just use yeast or bacteria all the time. It's not going to work. So we can tell with some of them, like if we need growth hormone or insulin or interleukins or bovine growth hormones or cellulase, all of these we can do with E. coli. There's some in there with I lost it. These are the yeast ones, mammalian cells for the three that we talked about. So we're going to need a variety of different types of cells, but we also still need to solve some other problems. So pharmaceutical researchers are currently exploring the mass production of gene products by whole animals and plants. Recombinant animals are difficult and costly to produce and may be cloned to produce more animals with the same traits. So that's where we got that whole Dolly situation. But in this particular case, this is a goat whose milk is producing insulin. So the whole idea is that this goat can kind of be genetically engineered to produce insulin in its milk that we can sort of get that protein extracted from. That helps the millions of diabetics that we have. Here's cloning, so you could think of the sheep Dolly. She was the first one to be cloned. That's a complicated situation, which is actually an interesting story. If you want to ask questions on how that worked, she didn't live very long, but you can ask. I'll tell you about clone of Dolly. So DNA technology, including gene cloning, is widely used to produce medicines and to diagnose diseases. Therapeutic hormones produced by DNA technologies include insulin to treat diabetes, like we just looked at, human growth hormone to treat dwarfism, again, tissue plasminogen activators to help reduce blood clots and heart attacks. DNA technology is actually used to test for inherited diseases, detect infectious agents like HIV. This one's actually the whole purpose of the glow cat, so that's one of the reasons if you've seen that one. Produce vaccines, some harmless variants or derivatives of pathogens to stimulate the immune system and have a lasting defense against the pathogen. So this is why we get our flu shot every year too, to try to avoid that. This was a year in particular, I was glad I got my flu shot. I was like, whew, that was a rough one. It was a doozy this year. So here's human insulin, and this is sort of an isolated version. That's what that is in the jar. And this is them sort of producing it on a larger scale. So here we go, a little bit more with our GMOs. So since ancient times, people have selectively bred agricultural crops to make them more useful. We will absolutely explore this in evolution. One of my favorite things to look at. DNA technology is quickly replacing traditional breeding programs to improve the productivity of agriculturally important plants and animals. So genetically modified organisms contain one or more genes introduced by artificial means. Typically, and I have asked this one because for me, I would take that a whole bunch of different ways. It's a little too loose for me. But I will promise that when we started doing this particular project years ago, I confirmed it with other scientists that I knew that it typically relates more with transgenic organisms, which means they contain at least one gene from another species. So when we look into those, and that could be simple things as like this has bacterial DNA in it, which honestly, it's like if you eat yogurt, you have bacterial DNA in your belly. But again, this is by artificial means. So this is, we're inserting the genetics of another animal or another organism into the original target organism, but not through means of like eating or whatever else. Like it's being incorporated into the genome specifically by humans. Okay, so that's a huge key point as you're doing your project. So GMO crops have a great many hungry people, improving food production, shelf-life, pest resistance, and nutritional value. Those are some of the ways that we've been able to help. There's been a great deal of research on golden rice, so it's a transgenic variety in 2000 with some daffodil genes. So we just have rice and daffodil that are kind of together. So if we actually kind of worked through that one individually, it might not seem as like something that might freak you out as actually talking about it. But it produces yellow grains containing beta-carotene, which our body uses to make vitamin A. So this is to help in countries where they have those kind of vitamin deficiencies. I don't know that the research has been around long enough to determine this. I've seen some research that says that it hasn't worked as well, but how do you know until you experiment? Hopefully, if you guys are doing golden rice and looking through this one, you'll be able to give us more information on that. So genetic pioneers are now creating plants that make human proteins for medical use. So these are human stuff. So pharmaceutical trials currently underway involve rice to treat infant diarrhea, corn for cystic fibrosis, safflower to treat diabetes, and duckweed to treat hepatitis. So although promising, no plant-made drugs intended for use by humans have been approved or sold. This is fun. We like to take lots of time in science, because when you move things along too quickly, that's when you have disasters happen. So they may move at a pace that's safe for human consumption. We dig that. We let a professional handle that. Okay, so agricultural researchers are producing transgenic animals by injecting cloned genes directly into the nuclei of fertilized eggs. So let's say if your thing is an animal, that might be something that you're looking for that it's doing. So genetically modified pigs convert less healthy fatty acids to omega-3 fatty acid, producing meat with 4 to 5 times as much healthy omega-3 fatty acids as regular pork. Atlantic salmon have been genetically modified to mature at half the time of conventional salmon and grow twice the size. Now, again, there's a whole bunch of ethical issues to explore on that one, but there's some other examples that you can kind of go from. Scientists use safety measures to guard against production and release of new pathogens. So to guard against rogue microbes, scientists developed a set of guidelines, including strict laboratory safety. And if you guys don't trust this, I can tell you all about having gone into a quarantine facility. It was no joke. So just ask me on that one, because I have been in a legitimate quarantine facility where actually I don't think the organisms are that dangerous if they're released, but they're so super strict on how they do that they're not taking that chance. It's quarantined. Ask me about it. I'll tell you all about it. So the genetic crippling of transgenic organisms to ensure that they cannot survive outside the laboratory, there is some of that in place. So some kind of genetic crumbling of it. A prohibition on certain obvious dangerous experiments. So today, most public concern centers around whether it's safe for humans and the environment. Again, I don't think there's a ton of stuff on that yet. It just takes a long time to kind of figure that out. But we may find it, because as a scientist, I can't say either way. Back in the day, they used to think DDT was super, super safe. It's a pesticide we'll talk about. I mean, they sprayed it everywhere. And then data showed later on that it was extremely dangerous. So we're in one of those areas where it's we're just not sure yet. We just don't have the data. I can change my mind in a minute as long as I get data that supports it in a certain direction. And that's what we're looking at here. So let's get the data, let's get the facts out, and let's see what we've got. Okay, so genetically modified organisms are used in crop production because they have more nutrients and are cheaper to produce. But do these advantages come at a cost to the health of people consuming them? We don't know yet. There was a 2012 study that involved 104 pigs that were divided into two groups. One was fed a diet containing 39% GMO, and the other one had a non-GMO corn. It actually showed that, I think in terms of growth, organ structure, and responses measured in 30 days and 110 days, that they didn't have any significant differences between the two, and no traces of foreign DNA were in slaughtered pigs. So there is some data to suggest that, but we might also think that this is only 110 days. You only over a couple months. So although the pigs are a good model for human digestion, some critics argue that human data are required to draw conclusions about the safety of dietary GMO in people. But you have to explore that. A human study of golden rice was conducted jointly by Chinese and American scientists in 2012. It concluded that GMO rice can indeed be effective in preventing vitamin A deficiency among children who relied on the rice as a food staple. So it's possible, but that's been a whole 5 years. So maybe we have more. Okay, so they tried capsule of pure beta carotene versus golden rice versus spinach, and it seemed like the golden rice was working better than spinach, and better than at least getting closer to a capsule. So, some thoughts there. All right, today no study has documented health risks in humans from GMO foods, and the general agreement among scientists is that they're safe. I don't think I have any data that conflicts with that from the last 5 years of doing this, but we could be wrong. So if you find some significant sources that say stuff and not just somebody's blog that says they're harmful, that's not a source, but a legitimate scientific study explaining how it's dangerous, then we'll need to know those. And some GMOs in particular are going to be dangerous. So we'll see that. We're going to explore all of that through these projects. So, we have that they're safe, but because it's still new, even 5 more years down the road still makes them just as new. So we still need more time to measure long-term effects of GMOs on humans. So concerns remain that transgenic plants might pass their new genes to related species nearby, which is kind of our ecological problem. So that's kind of what you're looking for if you need an ecological problem. And it might disturb the composition of the natural ecosystem. So whatever's going on in that particular environment. So you're looking for these types of things when it comes to your potential problems. This is sort of ecological, and the ecosystem gives us the environment. So look into those. So although the majority of several staple crops grown in the U.S., including corn and soybeans, are genetically modified, products made from GMOs are not required to be labeled in any way. And in 2012, the citizens of California voted down a ballot to measure requiring GMO labeling on food and drink in the state. We could have a whole debate on this one, but I'm just going to leave it where it is, and we can talk about it a little bit later. In the case of GMO crops, zero risk is probably unattainable, so scientists and the public need to weigh the possible benefits versus the risk on a case-by-case basis. So again, some that we read about in the next couple of days will be like, okay, and some will just be like, wow, that is really terrible, and that's fine. So we need to go case-by-case to make a judgment call. The best scenario would be to proceed with caution based on our decisions on sound scientific information rather than some irrational fear or blind optimism. So we really do need to go into it, and that is why we are looking for specific facts and data instead of just going by opinions or blogs. Let's see. Oh, actually, I forgot to take this out. This is my bad. But I'll leave it here for you, so if you're interested on it. This is more for therapeutic purposes of humans. This doesn't necessarily relate to your GMO project. It's just interesting as far as humans are concerned and how gene therapy we can use the gene from a healthy person and apply that to an unhealthy person and maybe be able to solve some of those issues. So there's an idea in those that we could talk about, but it doesn't pertain as much to your GMO project, more DNA technology. Okay, so you've got those. I'm going to leave it at that. If you have any questions at all or any concerns or any of this stuff doesn't make sense, shoot me an email from your personal email to mykellycroyce@gmail.com so I can give you some feedback and some help on this project. So hopefully this gave you a little background information. If you need more, just email me.